Okay, we're gonna move into chapter 5. Arguably the most interesting stuff, also potentially the most confusing, but it's some cool stuff. Okay, so evolutionary variation in proteins. So protein evolution, we're gonna talk about two key principles of it. One, variation. So variation is occurring at the level of DNA. It's changes to the actual nucleotide sequence in genes, okay? There's some difference in the nucleotide sequence of the genes at the level of DNA is how we'll define variation. This can occur from a couple things: errors in replication, some sort of genetic recombination, some sort of damage from the environment. Okay? So then the other piece of evolution we'll talk about is natural selection, and this is happening at the functional level of either RNA or proteins. So for proteins, we said there's a relationship that goes sequence to structure to function. So sequence changes that don't alter the structure or the function are tolerated, right? And we said there's a lot of redundancy, remember, in the genetic code, which means that there can be some changes without any dramatic, horrific changes to structure and function, all right? So sometimes there will be sequence changes that can be totally tolerated because they don't lead to any downstream changes. There are going to be changes that disrupt function, that want to be avoided. And then there will be changes that improve function. And so this is kind of where natural selection comes in. If there's some sort of improved function downstream, then these sequence changes might be adopted evolutionarily, like as you go down the line, okay? And those substitutions will start to happen more and more frequently. So just kind of remember that one of the things we're interested in this class is biomimicry. So if we could understand how and why and which sequences get adopted or avoided, then we could potentially intervene. So we could either intervene in some sort of pathological condition and try to correct it, or we could create some type of designer protein that has a very particular function that we want. But again, we have to go back and look at the sequence, look at the structure, figure out all the things that it does before we can start doing that.